Well, welcome everyone, and thank you for joining us for today's webinar, HPP Validation for Seafood Products, Ensure Food Safety and Extend Shelf Life, brought to you by Seafood Source and sponsored by Hyperbaric. Before we officially get started, I do have a few housekeeping notes to go over. If you have any technical issues using the webinar platform, we first recommend you refreshing your browser. That tends to solve most issues. But if you continue to have any technical difficulties, please use the Q&A box and I will respond right away. Note there's no call-in number for today's session. To connect, you just need to have access to either a computer, tablet, or cell phone, and everyone's lines have been muted. For your convenience, everything in the webinar console is fully customizable. So you can move things around, resize the windows, and even turn on captions for this event. There is a Q&A box where you can ask questions at any time using the Q&A box. We'll try to leave time at the end to run a Q&A. If we're not able to get to all questions, I'll make sure to forward that off to the sponsor for follow-up via email. And this, finally, this webinar is being recorded, and you'll receive a thank you email with the on-demand materials within 2 business days. This will include a link to the webinar console to log in again, to replay the webinar as needed, and download the PDF of the slides. And with that out of the way, I'd like to welcome, we have our 2 speakers for today. We have Dr. Alvin Lee, who is a microbiologist and at the Institute of Foods, he's the director, for the Institute of Food Safety and Health. And then we have Daniela Soto, who's the HPP Food Application Specialist at Hyperbaric. And at this time, I'm going to hand off the webinar to Daniela, who is going to be sharing her screen for her portion of the webinar. So please make sure to maximize your media box when she is sharing her screen. And with that, I want to welcome Daniela.
Thank you very much, Laura, and thank you all for joining us today in the Seafood webinar. As Laura says, my name is Daniela Soto. I'm an HPP Food Application Specialist based in Miami, Florida, where our USA office is located. I want to start introducing you to our applications team. This is a team of experts in the HPP field. In case you have any inquiry related to the applications of HPP technology or any other inquiry related on this topic, please contact us through the email apps@hyperbaric.com. So we are a team that is specialized on HPP. We provide technical support. We are dedicated to food science, and also we can provide help in packaging, product development, also guidance and validation studies. We lead what we call HPP pilot plants and incubators. So these are spaces in our facilities where customers can test their products, can validate their packaging. We have 3 incubators, one based in Miami, Florida, where I'm right now located, another one at our headquarters in Burgos in Spain, and another one we recently opened in China in Shanghai. And in these HPP incubators, we help customers launch different products. We provide 130 tests annually, and also we help customers understand their best processing parameters for HPP, also how to comply with food safety regulations, and also, again, provide guidance with validations and shelf life studies. Also, we dedicate a lot of our time in educating different customers or people who are curious in understanding how HPP works by giving this type of webinars or conferences, talks. Also, we attend different shows. For example, this year we will be attending Seafood Expo in case you would like to approach personally to us. And just finally about Hyperbaric, we're a company that have been around for more than 25 years. We're a medium-sized company. Also, we own 60% of the market share in HPP equipment installed, and we've installed over 400 machines worldwide. So now let me introduce you to what is HPP technology. So this is a technology that has been around for a lot of time, and it is considered a cold pasteurization technology because we will be inactivating microorganisms without the use of heat. And instead of heat, we'll be using pressure. So the pressure will be transmitted by using water as a pressurizing medium. And because of this, we'll be transmitting it isostatically, which means that it will be applied homogeneously throughout an already packaged product in its final packaging to help prevent gradients. So isostatically means that everything will be transmitted homogeneously and there will be no pressure gradients inside the product while being pressurized. So that is why it's important that the packaging you choose is flexible and also water resistant. Here, I brought you a video to show you how the technology works in our machines. Here you can see the already packaged product in its final packaging loaded into the blue baskets, which will then be loaded into what we will be calling the vessel, which is the place in which pressure will be transmitted. First, we start loading the vessel with low pressure water, and then we'll close it with the help of the plugs and wedges. After this, with the help of the intensifiers, which you can think about this as turbos in a car, the more you have, the faster you increase the pressure, we'll start pumping high pressure water into the vessel to help reach the desired final pressure. So each product that you process will have a different holding pressure, different holding time, and also pressure applied to it, depending on the type of application you're looking for. So here you can see how the intensifier works with the help of a piston that is moving to help increase the pressure. When the final desired pressure is reached, it will be held during a certain holding time. So this time will depend again on the product you want to process. After this time is achieved, then the pressure will be released and the vessel will come out of the machine with the already processed products coming out from the other side of the machine. So this is how an HPP cycle works. So you can see in this case, some guacamole cups were processed. So there are different applications that HPP has. Here I show you a graph where we explain the market overview of how it is distributed depending on the application type. As you can see, the juices and beverages lead the market, followed by the meat products, and then by guacamole, and followed by the tolling services, which are customers that have one of our HPP units and can provide the HPP service. And finally, the seafood products, which is a growing market for different applications. So I mentioned before that HPP can help extend the shelf life and also can help ensure food safety. And the way it is done is by applying different parameters of pressure and holding time, depending on what you want to achieve. So at low pressures, what we can do is we can inactivate deteriorative microorganisms such as yeasts, molds, parasites that can be in your product and also that are pertinent in different seafood products. So at lower pressures, we can extend the shelf life. At higher pressure levels, we can inactivate different bacteria and viruses. For example, Listeria, Salmonella, E. coli. And I would like to add that HPP cannot inactivate bacterial spores, so additional controls may need to be added. Dr. Alvin Lee will explain furtherly about this more in depth. So I want to touch more about the different applications of seafood products that HPP can help leverage. So depending on the application type, different parameters will be used, as I mentioned previously. So HPP can help automate shucking while also automate the meat extraction of different crustaceans, shellfish, and also bivalves. So this is done by applying the lower pressures. I'm talking pressure levels between the 29,000 to 50,000 psi, and holding times that are very short, from 1 to 3 minutes, depending on the applications type. At these parameters, we'll be able to reduce deteriorative microorganisms to help extend the shelf life, and at the same time, we'll be able to help deattach the meat from the shells or the meat from the lobster shell that has been processed to help extract it faster and also to increase extraction yields. On the other side, we have the application of HPP in different seafood products to help extend the shelf life and also to help increase food safety, either for cooked seafood or raw seafood. For cooked seafood, higher pressures that go up to 87,000 psi, applied between 3 and 6 minutes, will be applied. At these processing parameters, HPP can help inactivate pathogens such as Listeria, Salmonella, E. coli, which have been around different ready-to-eat products in different recalls from past years. And in raw seafood, HPP will need to be applied at lower pressures to help preserve its quality and also sensory attributes, because the way HPP helps denature microorganisms is by denaturing different key proteins inside of it. So again, as it denatures key proteins and microorganisms, it will also denature proteins in raw seafood. So we want to keep these pressures low to help preserve their sensory attributes at their maximum. I mentioned previously about the shocking application, which can be applied to different shellfish, bivalves, products. This application, HPP can help leverage and recover 100% of the meat of the products that are processed, either from lobsters, knuckle, claws, tails, also crabs. Crabs need to be tested to find the most optimum parameters. Also oysters, different oyster types from clusters, also like clams, mussels. They are compatible with the technology to help automate their opening, their shocking. Another application that I mentioned previously is raw fish. Here I show you two of our customers, Alcorta and Del Pierre in Europe, who process raw fish fillets in these trays with their film on top, and they use HPP to help extend the shelf life while using really low pressures. Also, another application that is going on is desalted cod. So this desalted cod, the protein is already denatured, so HPP won't change as much their texture or their color. So it is a very compatible application. Alcorta is a customer in Europe and Spain who is leveraging this right now. Another product type that is going around the market is cephalopods. So for example, pre-cooked octopus or raw squids, they're also compatible. As you can see, most of these products are packaged in trays, in films, either with 10K OTR films, which can also be compatible with the technology to help extend the shelf life while also preserving all the quality attributes of freshness that the consumer will be looking in this type of seafood applications. And I know that some of you who are seafood processors are looking on how to create value-added products in this seafood sector. So ready-to-eat seafood is something that has been going on around the market, and HPP can be a good solution to help leverage clean label products with an extended shelf life while also being safe from Listeria, mainly Salmonella, E. coli, these bacteria that are pertinent in these ready-to-eat products. So HPP can help leverage a 2-3 log reduction in it, and also can help extend the shelf life from 45 to 60 days while being stored refrigerated. These are some pictures of some customers' products that right now are using HPP as a post-lethal step to help protect themselves from mainly Listeria. Here you can see a product that has some carbs as base and then some cooked shrimps on top. Also, this customer provides to a cooked salmon fish on top, which is Listeria-free and also it is clean label. Another product is this cooked crab meat in cups, and also the white fish salad by Acme, which can also be processed with HPP. You can note that all these packaging options, they're flexible, they're water resistant. For the cups, it's important to consider that the process needs to be done without processing it through HPP without the lid, only the film, to help prevent any mold formation during the storage. Another ready-to-eat application are algae products. So this customer from Korea, they are processing this seaweed salad and in order to help prevent any Listeria contamination. They are using HPP as a lethal step to help achieve a 5 log reduction while extending the shelf life for 180 days by storing it refrigerated. Also, spirulina is also an algae that is compatible with HPP since HPP does not interact with covalent bonds, its nutritional value can be preserved. So this is just an value-added product made from guacamole based by adding in fresh spirulina. And now going more into shocking application, meat extraction. You may already know that traditional shocking is highly labor intensive, also very time consuming, and sometimes it can be difficult to achieve 100% of the meat recovery from these shocked bivalves. And again, some injuries can occur. So HPP is a great opportunity to help automate this shocking by achieving 100% of meat recovery from the bivalves or crustaceans you process. Here's an image of a lobster that was processed with HPP. You can see how clean the extraction was. Also, these oysters, they look more plump. HPP also caused this effect to, like, the proteins inside the meat, they get more hydrated. So that is why you get this plump appearance. I want to mention that for the shocking processes, the live product is loaded into the basket to help preserve its freshness. And also there are some studies that say that, for example, crustaceans in lobsters, they suffer less when processed with HPP. So the HPP process will kill them. So the live products are loaded into the baskets, and then they are processed through the HPP at really low pressures and reduced holding times. And then the automated shocking begins. So for oyster processing, the most conventional parameters will be between the 2500 and 2700 bar to help extract the meat easier and also to open those oysters, clams, mussels that will be processed. Also, HPP is a valid food safety alternative to help control Vibrio and achieve a 3.52 log reduction based on what the ISSC standard stands for, and also to help control norovirus. Also, HPP can help extend the shelf life depending on the product you're applying it to or the processing conditions you're applying to. Either you can reach 21 days without adding any preservatives. So for shocking bivalves, it's important to consider that you must find the most optimal processing conditions. So at really low pressures, you can see that no release can be seen. So you need to increase the pressure a little bit more. But if you increase it a little bit more, you can obtain broken shells. So it's important to find that sweet spot when processing bivalves. In this study, they found out that they had a total release while preventing the obtention of broken shells and preventing partial release of the meat from the shell by applying 275 megapascals for 3 minutes and 300 megapascals for 2 minutes. So what I do with customers is usually they ship me some oysters, I process it at different HPP parameters, and depending on that release of the meat from the shell or that opening, we decide the most optimum ones depending on their product type. So this is an example of some Pacific oysters, which you can see that there are clusters, which were processed just like that into the HPP machine. And we tried different processing parameters. This one was 2600 bars for 70 seconds. You can see how easy it was to just open the oyster and extract the meat from the shell. It just slides from it. Also, this cluster had other openings behind of it. You can see how also this one was shocked very easy and also how automated it was. This was another oyster cluster processed at 300 bars for 100 seconds. You can see how easier it was compared to the other one to just open the shell, open the oyster, and just get out the meat. How plump it looked, also how big it was, also how I was able to recover 100% of the meat. And finally, this parameter at 3,500 bar for 100 seconds, how easiest it was to just release the meat from the shell. So I mentioned previously that HPP can help inactivate Vibrio. Dr. Alvin Lee will explain more furtherly, but ISSC asked to achieve a 3.52 log reduction of these microorganisms. So here I show you just as reference different studies that have been done with HPP and different oyster products to help inactivate Vibrio. And you can see how at low pressures and reduced holding times, this inactivation can be achieved. And what ISSC is asking for can also be easily done. I mentioned previously that other bivalves can be processed successfully with HPP. This is a video done by our applications team. Here are some scallops. You can see how it slides from the shell, and also mussels and clams. There are other products that can be processed through HPP to help automate shucking. And just to give you some ideas on how you can market your shucked oysters with HPP, customers like to process already banded oysters through HPP, so they maintain closed during the processing. And after HPP, they are still open, but the band helps prevent the meat from falling off the shell, so it is still inside of it. Also, the half shell format is something that is very attractive because you have like this tray with little depths, and they just place the oysters already opened inside of it, and then they put on top film. Also, the vacuum pack one, where they extract the meat from the shell and then they vacuum seal them. And then either they commercialize it frozen or refrigerated. Another application for shellfish is lobster processing or shellfish. When processing this product, different processing parameters are used. So we will increase the pressure a little bit more, and we'll process at 2,000 for 4,000 bars to help deattach easier the meat from the shell and help recover 100% of the meat from it. Again, HPP can help inactivate Vibrio or norovirus in this. For this product type, there are different lobster formats which are compatible either from whole or split lobsters for like culinary aspects. Also, the raw or cooked knuckle and claw meat, which has been extracted already from the shell, and then it is packed into a 10 kOTR bag and commercialized. Also, raw or cooked lobster tails, there's something that is also common in the market. Our customers do that as well. And here's just an image of some companies who are leveraging HPP for shellfish products, either from the US and also from Europe. And just as closing remarks, as you can see, HPP is a very versatile technology which can help you create clean label products with an extended shelf life, while also ensuring food safety for different microorganisms of interest, while also automating shucking, which is something that right now the industry is really interested in, and help you open new market opportunities. So thank you all for joining us. We'll be presenting at Seafood Expo North America next week in Boston. And just for your reference, our booth number is the 2074. If you want to come around and meet us in person, we'll be pleased to see you there. Thank you. Now I'll hand over the presentation to Dr. Alvin Lee.
Okay. Thank you, Daniela. And good afternoon, everyone. Actually, I'm still in Pacific time this week. I'm normally based in Chicago, but I'm here at the IFTIPS meeting in Reno. So I'm in Pacific time today. But I'm definitely happy to accept the invitation here, and an honor to present some of our work here on high pressure processing of seafood. My name is Alvin Lee. I lead the Center for Processing Innovation within the Institute for Food Safety and Health at the Illinois Institute of Technology. The Institute IFISH is an FDA Centers of Excellence, along with 4 other universities in the U.S. My group predominantly conducts validation studies on processes, including novel technologies as well, and definitely high pressure with different types of foods, with different pathogens, including viruses. So, and our last project was actually on raw diet pet foods. So I've been working in the high pressure processing areas at least for the last 20 years, and I am still constantly learning new things and be amazed by what high pressure processing or HPP can actually do for foods, and also achieve some of the food safety controls that the industry have been craving for all the time, right? While still maintaining the quality aspects of the food. So I know Denal has presented a little bit on oysters and bivalves, right? These are Sydney rock oysters. These are work that I conducted while I was still in Australia at CSIRO. And usually at this time, I will not give you the answers. I'll just ask for a raise of hands as to, which you think is a high pressure process oysters, right? But for the sake of time, the answers are right in front of you. The one on the right are high pressure shucked oysters. The one on the left are hand shucked oysters. Sydney rock oysters are not very easy to open up because they're usually clumped together in huge mangled mess. And to harvest them, one would have to use pretty much a hammer to try and knock them off, and try and open them up as well. A lot of the Sydney rock oysters are actually sent overseas, and usually to faraway markets, right? So high pressure processing has actually helped in a way to reduce some of the transportation costs as well, but also extend the shelf life. And I'll show that to you as we move along. And obviously, with any processes that we deal with, well, we want to make sure that we maintain some of the quality, right? So this was done while it was actually in the U.S. already. So these are just Pacific oysters, and it was conducted through pressure. But also, we wanted to know how the quality actually looks like, right? Not only just looking at it, but also can we have some sort of quantitative measurement? So with this, we are using an eNOS to try and determine if there was any changes within the smell itself. The green line that you see is actually a control, and then the red line is one that we have processed, and we just use the highest pressure and time in order to conduct, one extreme over the other, right? So the green line is your controls, the red line is the high pressure process ones. And you can see that it actually mimics pretty much the same. Only thing is that the green line in certain areas are a little bit higher in the peaks, meaning that it has generated a little bit more of that smell that's characteristics of the oysters. And when we try to identify what those compounds are related to a fish light, to a fishy smell that some of the oysters panel actually felt that those were positive attributes. So I won't go into too much detail here, but it's just that certain characteristics of the smell from oysters were enhanced. Similarly, for the taste itself, we use the e-tongue as one of the analyses. And in this case here, oysters was processed in an increment from 200 to 600, an increment of 100 megapascals. But for the interest of time, let's compare again the green line plus the red lines. And you can see that the green lines are mostly clustered together in the middle, whereas the red lines you can see in certain aspects are towards the outer rings. And the further that it spreads out into the outer rings, meaning that it has that stronger taste. So for example, high pressure processed oysters had more of that umami taste that the panel actually quite liked it, right? So and this was then confirmed with the e-tongue that it had a lot more of that umami taste compared to your control oysters. Then, as I mentioned, that it can achieve some of the food safety aspects, but it also can achieve transportation through much further regions, right? So While I was in Australia, this was done together with a company that actually exports Sydney rock oysters. And we looked at in terms of high pressure processed oysters versus background counts. What would they achieve, right? Would they also achieve a shelf life extension as well? So let's start off with the right hand side, right, where we see coliforms. So if we process oysters and we leave them in boxes with moist paper towels over it, and then we open them up each day to try and assess for coliforms, and you'll see that in the controls where it's not treated with pressure, then coliforms shoots up after about 10, 12 days. Whereas pressure treated oysters maintain a pretty low level of coliforms, usually less than 2 logs. And then when you look at your standard plate counts on the left, you can see that the untreated oysters crosses that green line, which is an FDA standard, or the red line, which is the Australian standard for standard plate counts for oysters. So if we go by what that green or red line says, there's a slight difference of roughly about 5 days between standard plate counts for treated and untreated, right? So 5 days may not be a lot, but what when we worked with this exporter, what they told us was that that 5 days allows them another 5 days of shelf life, right? At the end, when the product actually reaches that destination. So in this case here, they can still ship them at refrigeration, but they still have that 5 days extra for retail sale, which was actually good for them as well, without compromising the food safety aspects of it. And they were quite pleased with it, is that now that they can actually ship Sydney rock oysters into say London in the UK, halfway around the world, and still achieve that extra few days of sale. So HPP in this way here can actually help in terms of extending that shelf life, but also at the same time to try and reduce some of the transportation costs, because in the past they probably would have to ship them frozen, and they lost also some of the quality when the product was then thawed out. So other than oysters, we have also worked on quite a few different types of seafood products as well. Certain seafood products, as you have probably have seen also from Danella, works really well, right? But also some do not really work too well unless you have something else to sort of mask some of that changes, right? So with shrimp and salmon, for example, treating it with high pressure processing can change some of the muscle fibers in there, that resulting it to have that slightly cooked appearance. Whereas with calamari and octopus and things like that, and squid, we don't really see a lot of changes in color, but also the texture as well. So just an interesting fact here on the salmon with the top left-hand corner ones. This was done quite some time ago, but when it was done as a way to try and establish what that quality was. And it was the interesting thing was when it looks cooked, and if you slice it up again into sashimi slices, it actually had that raw taste to it, which was a very weird sort of feeling and taste, like your mouth doesn't agree with what your mind is telling you. So it's kind of a strange feeling tasting those salmon pieces. So there are also quite a few references out there that sort of talks a little bit about the raw appearance and when discoloration becomes evident, right? So for various seafood products as well. So again, this is related to some of the muscle fibers within the seafood itself. So I worked with a muscle protein expert at that time, Ron Tume, who was a colleague of mine at CSIRO. So he did all these measurements here to look at, what really changes when we expose salmon to various pressures, right? So going from 150 megapascals to 250 megapascals. And what we are seeing here is the relaxation of the muscles, right? So like the myosin and the actin. And therefore, in the meat world, high pressure processing has been used as a way to also help to tenderize some cuts of meat as well. So I know some of you wanted to know in terms of now you have the quality, but then really what does it do to high pressure with high pressure processing and food safety, right? So let me start off with Juan Leon. He's at CDC, and when I moved to the US, we had a project called NoroCore, and the whole goal of that work was to try and establish controls for human noroviruses. That project has now ended, but then we have quite a lot of publication on the inactivation of noroviruses in oysters. So one at CDC actually did this into the seeding trials where he put live human noroviruses into oysters that we pressure treated, and then he fed this to volunteers and to see how many of them actually came down sick. So if you look at the let's say phase one at 400 megapascals, when it was treated at 25 degrees Celsius at 5 minutes, look at the untreated ones, we have 7 out of 15 that came down sick. With the high pressure treated oysters, 3 out of 5, basically 60% came down sick. But if you look at the third one, where the oysters were then treated at lower pressures, then we had a much more success in terms of lowering the number of people who came down sick with noroviruses. And then when pressure was then increased from 400 to 600 megapascals at 6 degrees Celsius for 5 minutes, actually, this was one of the more significant one where none of the people fed with high pressure treated oysters came down sick with human noroviruses, whereas 7 out of 15 still came down sick with the untreated ones. So high pressure processing can be one that can be used to inactivate human noroviruses. We then began to start to sort of look at in terms of how does high pressure processing and viruses or the different types of viruses behave, right, where they are subjected to pressure. So this is one studies that was done while I was still in Australia with my PhD student, Stephen Grove. He's now moved on to bigger and better things. And he generated all these curves, inactivation curves, on different types of viruses, right? So at that time, polioviruses, feline falciparuses were used as a way to mimic different types of viruses in foods. We also have the culturable strain of hepatitis A viruses that we use to try and see if hepatitis A, which was common in shellfish, could be inactivated by high pressure processing. So the long story short here is that feline falciparuses were very susceptible to high pressure processing, followed by hepatitis A, right? So you can see that it has that tailing effect when pressure increases. The interesting thing was polioviruses. To this day, we don't really know why is that resistant. We think that it's probably due to its configuration as a virus itself, in terms of the shape. Polioviruses tend to be more rounded, whereas feline falciparuses and hepatitis A viruses tend to be more isohedral and has got some sort of conformation to it, where there are angles involved and all that as well. So perhaps the pressure that was exerted on those angles compared to a structure that was round has an impact in terms of inactivation. So we went on further to sort of look at, okay, well, then if high pressure can be used to inactivate viruses, then what are the process parameters that we need to achieve in order to inactivate hepatitis A, and how does those process parameters actually impacts the inactivation, right? So in this case here, you are seeing 300, 400, 500 megapascals. And then on the left is 15 parts per thousand salt. And then on the right is 30 parts per thousand salt concentration. As you can see is that when salt concentration increases from 15 to 30, those inactivation now starts to shift to a little bit more to the right, right? So meaning that you require a lot more time. You also require a lot more pressures to actually achieve that inactivation. We think that the salt actually helps to stabilize that configuration of the viruses itself. That results in a little bit more of that resistance. So salt is one of those factors that are in play for process validation. Same thing with temperatures as well. So I know this sort of contradicts a little bit of what Juan was looking at as well. But what we found with hepatitis A, and it could also occur with other viruses, is that when temperature increases, then the inactivation of viruses tends to decrease, which is actually quite the opposite of bacterial inactivation, because now with the adiabatic heating that is generated by the pressurization process, generally bacteria actually inactivates at much higher temperatures, whereas viruses don't, right? So they like that cold temperatures in order to have that better inactivation. So you can see here from the blue line to the green line is a shift in terms of temperature, and therefore there is also a change. Similarly, the matrix itself also can impact on the efficacy of the inactivation. So studies that are conducted in homogeneous, studies that are conducted in buffer has values in it, but then when it comes to the real thing, be mindful that there can be some change in terms of that inactivation. And with this one here, we are seeing temperature and seeing homogeneous and whole oysters having that slight difference in inactivation. Sorry, I got ahead of myself just now. This is the effect of temperature. And it goes from the left is 4 degrees Celsius, and the middle one is 10, and the next one is 20 degrees Celsius. This is an example here is just done in buffer, just to demonstrate that the effect of temperature results in the shift of the inactivation curve towards the left. Towards the right. And then, we have already established that pressure can be used to inactivate Vibrio. Pressure can also be used to inactivate viruses, right? So how do we wrap things up together? Because with seafood hazard, the FDA has already indicated that certain types of seafood can be prone to contamination or presence of certain types of microorganisms. In particular, Listeria monocytogenes, which we already know that can be inactivated by high pressure. Clostridium botulinum is another one that Danilo has already mentioned that is a spore former. It doesn't get inactivated by high pressure. But then there are also some studies that shows that pressure can sometimes inadvertently activates the spore, and then resulting in germination of the spore to vegetative cells and ultimately the production of neurotoxins. right? So the controls for Clostridium botulinum is actually very well defined within seafood hazard regulations and the guidebooks that FDA has produced. So ways of controlling Clostridium botulinum one is pH, right? So that's why a lot of our foods are acidified, is to reduce the chance of Clostridium botulinum germinating and growing and producing that toxins. So in general, a pH of 4.6 or less is defined as acidified and non-acidified foods. You could also try and use salt as a way to bind up some of the free water so that you reduce that water activity, reduce that moisture. Salt, sugar, or any ways in terms of removing moisture from the product can help in controlling Clostridium botulinum. The other control, obviously, is pasteurization, right? So with seafood products, we're generally dealing with non-proteolytic Clostridium botulinum. And there are a lot more heat labile than your proteolytic form. The proteolytic Clostridium botulinum is usually associated with heat-treated products. They are more pronounced in terms of when something is used in canning industry, for example, that has been treated with high temperatures because they are able to survive a lot better at a higher heat. Whereas non-proteolytic Clostridium botulinum are a little bit more labile. However, they can persist and grow at very low temperatures, which is more related to what seafood products are usually kept and stored. So the other control sources could be harvesting. So this is usually applied more to molluscan shellfish as to when you harvest that shellfish can reduce that microbial load and ultimately the amount of Clostridium botulinum that might be there, right? So controlled harvesting is one. The only way that we can use temperature controls here for non-proteolytic seaboard is to keep that storage temperature to well below 3.3 degrees Celsius. So other than that is your heat or some sort of pasteurization, and then before you pack them into reduced oxygen packaging, but then you also have to prevent some cross-contamination from happening, right? So the control guides and all that actually has defined all those processes within the hazard seafood guide. Then, this just summarizes some of the risks that we talk about, right? So that the neurotoxins can be produced at a when conditions are favorable for them. So non-proteolytic seaboard, usually, if you look at the literature, there will be types B, types E, and types F, because they are able to grow under refrigeration conditions, and toxins form much quicker when they're subjected to high temperatures. But usually non-proteolytic bot don't grow very well past the 26 degrees Celsius, right? So that will be probably one of where they're optimum, but it's also not the optimum in terms of where we keep seafood, right? So therefore, there's a temperature relationship. So the lower the temperature, if seaboard was to grow and produce toxin, it will take a bit of time to actually grow. So therefore, the longer the shelf life of that product, then there's always that increased risk in terms of when non-proteolytic seaboard may produce toxins. There are also documentations out there that talks a little bit about reduced oxygen packaging with cooked product, and that a temperature of 4.4 degrees Celsius is sufficient to control your proteolytic one because they are not going to grow, right? But then also your cooking process kills some of your non-proteolytic ones. So you have sort of controlled that risk from seaboard. But now that concerns now with that cooked product now changes over to Listeria monocytogenes. And then there are also some studies that sort of indicate that high pressure processes can sometimes inadvertently trigger spore germination and there are quite a few out there. And in fact, FDA has also done some of their own studies as well to actually show that high pressure is one that triggers the germination. So we have to be quite careful in terms of how high pressure has to be applied. It has to be applied with some sort of controls so that it doesn't go on to continue on its germination process. The other controls that we have been trying to tinker with and still continue to have that discussion with FDA is is that seabed do not actually grow under aerobic conditions. So therefore, if we can keep a high pressure treated process package under aerobic conditions, then perhaps that might be a way to stop seabed from growing, and also perhaps one of the parameters for controls as well, right? That we can use for the for seafood products. So raw seafood products and refrigerated reduced oxygen packaging challenges, right? So I just listed a couple of combination here. And if you have a sole barrier of refrigeration at 3.3 degrees Celsius, then if you look at the seafood hazard guide, it tells you that you require a time temperature indicators, right? A TTI. So right now, if someone was to use high pressure processing, and then on seafood, the recommendation here is to use TTIs, right? So I know it's a hassle to use TTIs, and it can be also another cost as well, right? So not only with the high pressure part of it, but you also have TTI use as well. So and it can be quite can be not feasible in terms of what the economic side of things would be. So then the other option would be to freeze it, right? So and also freezing products sometimes faces some sort of challenges to because of that quality, right? So if the product is frozen, then you have to have some sort of indication that the product needs to be maintained during frozen storage at retail. But if none of the above and you decide to use high pressure processing, then FDA or under the seafood hazard guide says that the inoculated pack studies done with moderate abuse conditions needs to be conducted so that you could validate the controls. You could validate also that non-prolific seabed is not going to produce toxins. And I'll explain this a little bit towards the next few slides as well. So let's say if someone was to use high pressure processing, has about 3.5% water phase salt, and it's also antimicrobial, then in this case here, an inoculated pack studies would be warranted. And also generally, FDA, even though seafood hazard guides don't really say that, but between FDA and myself, we always try to advocate for someone who's using controls to have at least two controls where possible, right? So if one fails, then you're not gonna, you still have another controls. But if you're using one barrier or one controls, then if that fails, then your product could potentially be spoiled. So in the seafood hazard guides, it talks a little bit about 10K OTR films, right? So I'll just put it into a cartoon here to try sort of explain, why FDA says that, well, if you're using a 10K film and with high pressure, then an inoculated pack studies would be needed, right? Even though we could argue that the 10K film still has some sort of oxygen transmissibility. So on the left side here, without high pressure processing, that 10K film has some sort of oxygen transfer between the inside and the outside, right? So the pack stays aerobic. seabed doesn't like it, but your spoilage microorganisms likes it. So it will increase over time, and therefore it controls also the amount of Clostridium botulinum that might be in that packaging itself. Therefore, the product is going to spoil a lot quicker before CBOT has the opportunity to actually produce toxins. So as a consumer, you could see that the product is visibly spoiled and you're not going to buy that product. So the thinking here, and also in a sense, is logical as well, and is that FDA says that after high pressure processing, do we still understand if that transmissibility of oxygen between it within the 10K film is that still maintained, right? So is the pack going to stay aerobic? But if not, then CBOT will like it, right? Because now it doesn't have that enough oxygen in order for it to control CBOT. Secondly, is that high pressure processing decimate a lot of your spoilage microorganisms that's used to control the CBOT within the product itself. So now, in the absence of spoilage microorganisms, then CBOT is going to flourish, right? So, and we can't tell if neurotoxin is produced because it's colorless and it doesn't tell us exactly when the product is spoiled as well, right? Because of the lack of spoilage microorganisms. So right now we have extended shelf life, but we cannot tell if the product was spoiled and spoiled because of spoilage microorganisms, or the product was spoiled and not fit for human consumption because of the production of neurotoxins that might be there, right? So, so now we have created an environment that potentially CBOT can actually grow, and therefore we need to use some sort of inoculated pack studies in order to verify that those conditions do not result in neurotoxin production. So, and this is all documented within that Seafood Hazard Guide as well, in chapters 4 and chapter 17. But then there are also languages there to sort of talk a little bit specifically about high pressure processing, and that the use of 10K OTR films, for example, on the bottom left corner, may not be suitable in order to, for that film to actually maintain that aerobic conditions within the package itself. So, and therefore, in conclusion here is that high pressure processing can be effective in reducing the spoilage microorganisms and extending that shelf life. But when CBOT is the target, then HPP can still be used, but it has to be used with further evaluation, like in conjunction with TTIs or in conjunction with inoculated pack studies if other controls are involved as well, because the risk from non-proteolytic CBOT from that use, right? So, but it is still a very good process to actually control different types of pathogens within the seafood itself. So with that, I think I've taken up most of my time, but if you'd like to sort of discuss this a little bit more, my email and phone number is on this slide here, and we are always happy to sort of help industry out in terms of their applications with high pressure processing. And I'll revert back to Danella to see whether we have enough time for question and answer.
We have time for one question that we got in, and Daniela mentioned in your presentation that the oysters looked plumper due to hydration during the HPP process. Is there a significant amount of water that is added? Is this something that the customer should be informed about?
No, usually this like weight addition is between like 15 to 20%. It ranges like that. And usually the liquors around that oyster meat is, you can think, like absorbed by the meat, because HPP will promote proteins hydration. So that is why you'll see some gain weight after processing it with HPP. But yeah, also they look shinier. I've seen also like more plump too.
Yeah, from our work in the past is that the high pressure actually denatures the two adductor muscles that keeps the shell intact, right? So when you're shucking the oysters, you tend to injure or tear the adductor muscle. So therefore, the insides of the oysters do break, and ultimately liquid comes back out. But when the adductor muscles are intact, then the oysters remain as it is. So and definitely looks a lot better than the hand sharp oysters.
Great. Thank you very much. We are out of time. I do have a few housekeeping notes to go over before we officially close out. As a reminder, the on-demand materials will be emailed to everyone in the next 2 business days. This will include a link to access the webinar console where you can replay the webinar and download the slides. Also, if you could please fill out the short evaluation form that will appear on the webinar console shortly. Your feedback is greatly appreciated. I want to thank Dr. Lee and Daniela for a great webinar today, and I want to thank all of our attendees for taking the time out of your busy day to join us. This webinar was brought to you by Seafood Source and sponsored by Hyperbaric. Thank you all and have a great rest of your day.
Thank you.